Thanks to Bob and other organizers for inviting me to share some of our work, which is in collaboration with, as Joan mentioned, both with her as well as Bob Rowland's group, as well as other collaborators on the host response to PRRS, the genetic basis of host response to PRRS. So last talk we talked about the virus side, and we've heard quite a bit about the virus side. Now we're going to talk about the genetics of the pig and how it is related to infection with PRRS and how the animal deals with PRRS. Well, acknowledged by co-authors, I don't have to introduce you to PRRS, but I do want to talk to you about, we know there's different strategies to deal with PRRS in the industry, and so what we're going to focus on is how can genetics of the pig, how can that contribute, can be one of the components of a integrated strategy for control of PRRS. And we think there is a role, and that's what I'll be talking to you about, and that's what we've been working on. Okay, and this all started in 2007 with Joan, Bob, and Jim in particular, starting what's called the PRRS Host Genomics Consortium. And the impetus was new technologies, new genomic technologies that came on board, in particular being able to do high-throughput genotyping of lots of animals for lots of genetic markers across the genome rather than having to look at one marker at a time and developing one marker at a time. And that consortium was developed with sponsorship from the Port Board in particular and USDA and very strong industry collaboration. And over time, so now we're in 2014, this consortium has grown into a even broader consortium involving multiple countries and many industry partners, different sources of funding, and many scientific collaborators. And so it started all with the initial PRRS Host Genomics Consortium, nursery pig infections with PRRS, which I'll talk about, initially funded by Port Board and USDA, but then since then the second version of that funded by Genome Canada, and I'll talk about that second version also. At the same time, Daniel Chibano at Nebraska has been working on experimental infections of pigs with PCV2, also in part funded by NIFA and Genome Canada and Genome Alberta. And then more recently, we have moved closer to the field, or models of the field, where we look at co-infection. And the model of co-infection that we've been working with is co-infection of PRRS and PCV2, to more closely mimic what might be going on in the field. And at the same time, we are also starting to use field data, where we bring clean piglets into health challenge facilities. Of course, this is all on the growth, focusing on the growing phase, but we know there's also a big reproductive component to PRRS. And so at the same time, funded by Genome Canada, John Harding has done experimental infections of pregnant gilts. I won't talk about that in this presentation, but quite a bit of work has been published already. John Harding was here, but he left. He had to catch a plane. And also reproductive information from the field, funded by PigGen Canada, which is a consortium of breeding companies that have nucleus facilities in Canada. Many of the PRRS Host Genomics Consortium companies are part of that, and funded by the Canadian Swine Health Board. And I'll talk to you about results from that project also. So quite a number of different projects, all categorized by large numbers of animals for the most part, and deep phenotyping and utilizing genomic tools to be able to look at the DNA and gene expression, RNA. In the next talk, Joan will be talking about more the transcriptome aspects of these projects. So what I'll be talking about is primarily the SNP genotyping results, so doing genome-wide association studies. Joan will be talking about transcriptomics, but in addition to that, there's also been work that's ongoing on proteomics, genomics, and also looking at in vitro assays. This is generating a huge resource in terms of samples, tissues, and data that will all be available and is used for different purposes. They go into project databases, and the outcomes are identifying regions of the genome that are associated with response of pigs to infections, doing what we call genomic predictions, trying to predict the performance of animals based on their genetics, their genetic markers. Also used by people at the Roslin Institute in the UK to develop models of PRRS infection dynamics that will be very useful to look at different aspects. And hopefully we are also able to identify improved vaccine targets. So lots of partners involved. I've already mentioned that, industry partners, funding partners, and lots of scientific collaborators. Iowa State, a lot of kudos to Kansas State University, Bob's group, and Maureen, Ben, and Becky, and others involved in the tremendous amount of data that they collect. And I think they're up to over 3,500 piglets that they've infected as part of these trials. University of Alberta, so Graham Plastow has led a lot of the Genome Canada projects. Jones Group at USDA doing a lot of the assays and also storing a lot of the tissues and the samples and being involved with arranging genotyping. John Harding's group doing the gilt, the gilt pregnancy, the pregnant gilt experimental infections. Roslin Institute, and then more recently also the University of Minnesota has become involved. Okay, so starting with the initial experimental infection models, the nursery pig challenge model that was developed by Bob Rowland at Kansas State. The idea is to bring groups of 200 commercial piglets into their facility, and then the initial trials challenged them with one particular strain, the NVSL strain of the PRRS virus. And so they are brought, right after weaning, they are brought into Kansas State. They have 1 week to acclimate, and then they're all infected because we want to look at between animal variation. So they're all infected, and then followed for 42 days, looking at weight, looking at serum to look at viremia, and then also getting tissue at the end to be able to genotype them using the high-density SNP genotyping platforms. The first results that we published, and we've published 3 papers on that, did analysis of the first 8 trials, and each of these trials, for the most part, represents different genetics from commercial breeding companies. Here you can see the parental breeds and both males and females being involved in these challenges. Typically, we see these kinds of growth curves starting from the point of infection, quite a bit of variability, some drop out. And this then shows the viremia, PCR-based viremia on the log scale. So all piglets get infected, and then they start to clear the virus. We do have this rebound, which we believe is more due to the virus changing or escaping the immune system rather than the genetics of the pig. And so for a lot of our work, what we focused on is the first 21 days after infection, which we call viremia, or viral load, which is the area under the curve. So that's the trait that we are looking at. When we look at the heritable components, we find that both growth rate and viral load are quite heritable, or moderately to highly heritable for the types of traits that we're looking at. We are very surprised, not surprised, pleased with the high heritability of viral load, so that indicates that when we look at the variability between animals, that over 40% of that is due to genetic differences. We also look at the correlation between growth rate and viral load. It's negative generally, and more negative at the genetic level than at the phenotypic level. So when we do these, so all these piglets are genotyped using 60,000 genetic markers across the genome, and then we do our analysis to find which regions of the genome or which genetic markers are associated with differences between animals in growth rate and viral load. And this is a so-called Manhattan plot, where each chromosome, or actually these are 5 SNP windows, but each chromosome has a different color. And then the y-axis indicates the proportion of genetic variance that is explained by that window of the genome. And immediately what jumped out was this region on chromosome 4 that had a large impact on differences between animals in viral load. And it explained about 15% of the genetic variance. So remember, viral load, about 40% of the variance we see in viral load between animals is genetic, and of that, 15% is explained by this region, which we in genetic terms would call a major gene, a gene with a major effect. And it's quite remarkable, especially given the different genetics that is represented in these analyses, that we, across these different genetics, we find such a strong effect. When we look at weight gain, we find that same region shows up. It's exactly the same region. And in that region, there is one SNP that we call our tag SNP that tracks the genetic differences that we see in there. Now, we're not saying that this is a causative mutation. We actually think we have found the causative mutation. This is a region that has quite a number of strong candidate genes, including members of the GBP family. And Joan will talk a little bit more about that, but through transcriptional analyses, we've identified GBP5 as the likely culprit of this effect, which is a gene that's involved in the innate immune system. When we look at this tag SNP, so when we look at the more details, so there are 3 genotypes for that SNP, the AA, AB, and the BB animals. The B allele is the favorable allele. And we don't, it's typically at lower frequency, so we don't have a lot of BB animals, only 44 out of the, out of the over 500 that are represented here in these first 8 trials. So we primarily look at the difference between the AA in red here and the AB. And you can see that the AA animals have higher level of viremia over those 21 days than the AB. So the AB piglets are able to clear the virus more quickly. And when we look at the individual trials, comparing the red versus the yellow, the AA versus AB, it's a pretty consistent picture, which again is quite remarkable given the different genetics that's represented. And then when we do the same analysis for weight gain, so now the AB piglets, the favorable genotype, has higher weight gain. And again, that's replicated across most of these trials. Looking at a little bit more in terms of dynamics, so this is the average curve for the level of viremia on the log scale for the AB pigs in red and for the AA pigs in orange or yellow. And you can see that, well, the differences don't seem very large, but they are on the log scale. And they are very highly significant. The p-values are up here. And when we then also look at weight gain, you can see that the differences in weight gain, they lag behind when we see the differences in viremia, but about a 5 pound, 2.5 kilogram difference in body weight after 42 days. So quite a remarkable difference. Of course, this is one virus, one strain of the virus. So the logical next question was, well, is this same region also protective when it comes to another strain of the virus? And so the next set of trials funded by Genome Canada and Genome Alberta were done with the KS06 strain of the virus. And so this again repeats the results we had for the NVSL for viral load, so the AA having higher level of viremia than the AB, and for weight gain, the AA having lower weight gain. When we look at the KS06 for viral load, we see the same effect, okay, with the AA having higher level of viremia than the AB. So it does seem to be consistent in terms of the effect of this region on these 2 strains. For viral load, when we look at weight gain, the trend is still there, but it's not significant based on this analysis. And that may be a result of the difference in the level of pathogenicity of the KS06 versus the NVSL strain of the virus. But nevertheless, there does seem to be an effect on level of viremia of this region. So the next question then was, well, okay, it works for PRRS-only infection. What happens if you have co-infection, which is more typical in the field, as we all know. And so we were able to get a NIFA grant, a translational genomics grant from NIFA that has the research component, but it also has an outreach and education component. And the outreach and education in particular targeting the veterinary profession in terms of working with them on the potential importance of animal genetics. So these co-infection trials also look at vaccination response. So the other question we have is, are there genomic regions in the genome that make some pigs more responsive to a vaccine? And here we're talking about a PRRS vaccine versus other animals. So these are, again, groups of 200 piglets. We actually split those into two rooms, one room that is vaccinated and the other room is not vaccinated. The other thing we do in those trials is, in order to get more power to look at the chromosome 4 region, we pre-select them to be AA or AB. We find the B allele to be dominant, and so and also because of the low frequency of BB, we don't look at the BB genotypes, but we compare the AA versus AB, and then so it's a 2x2 design. So these piglets, they come into Kansas State again after vaccination or after weaning, and half of them, one room is immediately vaccinated with a modified live virus of the PRRS strain, or PRRS modified live virus, and then 28 days later, all piglets, both rooms, are co-infected with field strains of PRRS and PCV2 that were isolated from a herd in Kansas that showed PCV-AD. And then they're again followed for 42 days. So the typical results we get, so these are the results for PRRS viremia on the log scale. So these are what you see here are the individual curves for the vaccinated pigs. The non-vaccinated pigs are, for the most part, don't show viremia, PRRS viremia, and of course this is the vaccine virus that is replicating in these pigs. And then and this is what happens after co-infection. So you can already see that the vaccinated pigs tend to have lower level of viremia after co-infection than the non-vaccinated pigs. So looking at that more statistically, and this actually was shown in posters by Janelle Dunkelberger from Iowa State and Megan Niederwader from Kansas State. We do find there's a relationship between the level of viremia, area under the curve, after vaccination, and area under the curve after co-infection, a slight positive relationship, so indicating that pigs that show less viremia as a result of the vaccine also tend to show less viremia as a result of co-infection, subsequent co-infection for the PRRS virus. When we look at after vaccination, the effect of the chromosome 4 region, AA versus AB, we don't have huge number of animals here. There's only 2 trials at this point. We'll be getting additional trials, but it is borderline significant, with the AB pigs showing lower levels of viremia for the vaccine, also for the modified live virus. And then when we look after infection, so this is viral load for the first 21 days after co-infection. We see that, both vaccination has a significant effect, comparing these versus these. So the vaccinated pigs show lower levels of PRRS viremia, as we can see from the graphs. But also the AB pigs, they tend to have, actually that's highly significant, they have a lower level of viremia after co-infection also for both the vaccinated pigs and the non-vaccinated pigs. When we look at growth rate, the results are, these are the graphs again. When we look at growth rate prior to co-infection, we see that there is an effect of vaccination. So the vaccinated pigs grow slower than the non-vaccinated pigs. So there is a definite impact of vaccination on growth rate right after vaccination. And there is an effect of the chromosome 4 region also, that certainly when they're vaccinated, more clearly so, there is a higher growth rate of the AB pigs than the AA pigs, consistent to what we've seen in the PRRS-only challenge studies. Then when we look at after vaccination, so growth from day 0 to 42 after vaccination, we don't see a significant effect of either vaccination of W or WUR. So these are still preliminary results based on limited data. There are some trends here with the AB pigs growing a little bit faster with the non-vaccinated, but opposite in the vaccinated group. So the jury is still out on what's happening there. We did see an interesting result when it came to vaccination. So these are 2 trials, and the impact of vaccination on growth from day 21 to 42, so the later part after the later period after co-infection, that we had an opposite effect of the impact of vaccination. So in the first trial, trial 16, the vaccinated pigs growing slower, and in the second trial, the vaccinated pigs growing faster. Although these are, again, borderline significant, but the interaction is significant. So more data, again, needed. When we look at PCV2 viremia, and this is all basically starting at co-infection, which is where we have that data, very noisy data, a lot of variability. But when we look at area under the curve for PCV2 viremia, we see that there's no impact of vaccination, but there does appear to be some evidence of an effect of the chromosome 4 region, with in particular in the vaccinated animals, so the animals that were vaccinated for PRRS, the AB pigs showing also some protection for PCV2 after co-infection. We also have field trials that I mentioned, where the approach is to bring clean piglets and take them into health challenged facilities, and these data are still coming in, and so I won't be able to show you any of the results of those, but and there they followed all the way to market. So the next thing I want to talk about is what happens on the reproductive side, and again, I'm not going to talk about John Harding's studies, but I'll talk about the field studies that we've been involved in. And the idea there is to take clean replacement gilts from multiplier farms, from different genetic sources, high health status, and take them into commercial herds that typically have a low health, or herds that were chosen to have a low health status. A lot of different genetics again, a lot of different environments across Canada, herds across Canada funded by the Canadian Swine Health Board and PigGen Canada and Genome Canada. Again, collecting lots of samples and looking at different aspects of response, and all that information going into resource databases, and trying to get at outcomes in terms of genetic cell selection tools, early indicators of health and performance of reproductive sows, and building a collection system and a data resource and a tissue resource to be able to go back to do further investigations. One of the things that came out of this is that as the companies were trying to source these genetics, these clean genetics, one of the multiplier herds broke with PRRS, and so we made use of that opportunity to study that herd in detail. This is they had a lot of very good data collection. So these are the rolling herd averages for born alive, number born alive at 24 hours, and you can see that rapid drop right around this time period in number born alive, and the increase in number stillborn and number mummified. And then based on that, we approximated that this was the date when the outbreak occurred, and we mobilized our veterinary resources, or the company mobilized their veterinarians that were working with that herd, and they were able to get the veterinarians into that herd 46 days later, after what we think was the outbreak, to collect blood samples on all the sows in the herd. And And what we did then is use the data to estimate heritabilities for the different traits. The blood samples were used to do an IDEX ELISA for PRRS, specific ELISA test, get an SP ratio, and quite remarkable, the SP ratio, which of course we get after the PRRS outbreak, had quite high heritability. So the level of the variation we see between animals in level of antibody for PRRS, about 45% of that is heritable. The other interesting thing is that when we looked at the genetic correlation of that antibody level with reproductive performance after the outbreak, we got quite high genetic correlations, and most of them being in the favorable direction, meaning that if we have boars whose daughters have high level of antibody after the outbreak, that those boars also have daughters that have a better reproductive performance during the outbreak. Okay? And so we think that this may be a potential way to select boars that have whose daughters will have better are better able to withstand a PRRS outbreak. More data is needed because the genetic standard deviations are quite large. And then we genotyped them all and did a genome-wide association study. And quite interesting, there are regions on chromosome 7 that show quite a large proportion of genetic variation in antibody response. And several of these regions are right in MHC. So that was quite interesting also, and these are large effects. So that was one of these herds, but then we got all the data from all the other herds also, and again did genetic analysis. So we are able to get SP ratio on large numbers of gilts, about approximately 40 days after they entered these health challenged facilities. Some of them came in with vaccination for PRRS, so quite a large mixture of types of environments, types of data. About 83% of the animals tested positive for PRRS at that time when the blood samples were taken. But again, this was quite heritable. So despite the messiness of the data, we can detect a very strong genetic component to variation in antibody levels approximately 40 days after entry into those health challenge facilities. Then also we did and this was done by Nick Sarau, who is one of the postdocs in the group, did a genome-wide association study, and again found these regions on chromosome 7 showing up. So the same ones that showed up for the outbreak, we also show up when we look at the gilt acclimation. Quite different circumstances compared to the outbreak situation. So one of the questions we then had is, well, can we use the estimates of the marker effects that we identify in the gilt acclimation data to predict what the SP ratio of animals are in the outbreak herd? And one of the things we did was to use genetics that was quite different. So this is the principal component analysis of the genotypes of the animals in those different herds. These are the gilt acclimation data, and this is the outbreak data. So basically what we're trying to do is, well, can we use this genetics to estimate effects of markers across the genome for antibody level and use those estimates to predict the antibody level of animals of this genetics in an outbreak herd. So quite different genetics and different situations. And what we find is when we do that, we can predict these genetics for antibody response with an accuracy of about 58 to 68%. And most of that predictive ability comes from the MHC region. So this says that we can use genomics to identify pigs that have higher antibody levels, per specific antibody levels with reasonable accuracy. And our earlier work in terms of genetic correlations show that this is also, that antibody level is also predictive of subsequent reproductive performance during an outbreak. So there's some promise that we will be able to utilize some things there. So conclusion, so piglet response has a sizable genetic component, piglet response to PRRS challenge. We have an important region on chromosome 4 that controls that, but there are other regions across the genome. Most of those effects are fairly small, however, but we may be able to use them for prediction, identify superior genetics. Reproductive PRRS, I showed you some of those results. Still a lot of work is ongoing and will be coming forth in the next little while. And when we look at antibody response, PRRS-specific antibody response, it is also heritable and maybe a good genetic indicator of reproductive performance during a PRRS outbreak. The MHC is an important contributor to that. And so the final conclusion is that we believe it is possible for us to select animals that have improved host response to PRRS, and that can be one of the components of fighting PRRS as an industry.